I'm very glad that I can present today here. And if I say I, then this includes Fatoni Musiafa, because he is the person who has been doing all this work essentially in a so-called INF project within the SFB 1208. And the reason why I'm glad that I can present this here is, on the one hand, we use eLabFTW, but we also somewhat, let's say, reworked it, not the eLabFTW itself, but the way you can work with it. And we would be very happy to get feedback from you on this, on the one hand, and on the other hand, we have been doing this as part of the INF project so far, and we would like to outreach to NFDI initiatives, and obviously the one for chemistry is clearly on the top of the list here. In particular, I would, in the remainder of the talk, you will hear about a tool that is called LISTER. And the overall idea that we had when we started this work was that we needed to use components that were more or less given at Heinrich Heine University to establish a research data management workflow. And when we thought this through, it was not thinkable for us that PhD students and postdocs, after 2 or 3 or even 4 years work in this SFB, will be gladly going back to all their data and assign metadata to it. And so we wanted to have a process that does this online, if you want, or inline, I should even say, in a semi-automated way. And that is the outline of the presentation. So one can say, what is the motivation? There will be not too much motivation, you're all aware of this, but perhaps many of you have seen this paper from 2016, where 1,500 scientists acknowledged that there is a huge problem in reproducibility. And as a chemist by myself, I was thinking that we do quantitative science and that this should be not so tremendous in our case, but apparently from what you see here, chemistry perhaps is at least as involved as other disciplines here. And then what you usually hear, and particularly you hear this from administrations, is you only have to store data according to FAIR principles, because that will solve everything. And we believed in this at the beginning until the point when we started to ask, what does it mean? And as an idea, I take a reference from a different field, machine learning. I would say this is a rather young field, a field where people are used to work with very large data sets. And they, in 2021, published a suggestion for reproducibility standards. And I find it very interesting that they do not only consider data models in source code, for example, as a requirement to do this, but for actually getting from a findable and accessible data to one that is interoperable, you at least need to specify the dependencies. But the gold mark you only get if your data is reproducible. And they require this, in the essence, by a single command that more or less press a button, be able to reproduce the numbers that have been published in the paper. And that is what FAIR could mean. On a more, let's say, local scale, we thought about use cases at the lab level that we have. And obviously, these are questions such as, can you collect data for thesis or publications after 2 or 3 years working in the area? Can you prepare summaries for reports or preliminary results? Can you find and filter data from fellow lab and ex-lab members? This may be more complicated. And perhaps even that we need to do this after years, or in a stress situation, if, for example, you're accused with fraud in a publication. And then on the obviously medium-term run, you would like to associate your data with external data repositories. And I will show an example later. So the requirements that we identified were, we of course have to have access to primary data. This is then where eLabFTW comes into play. We need to be able to extract metadata, pool both for long-term storage, and then link this to existing ontologies and perhaps knowledge bases to put the data into a context. And obviously, I'm aware in the chemistry field, or in my field of chemistry that I work in, there are databases where you can submit structural information of biomolecules, for example, or of small molecule data. Steffi Weidkamp-Peters, who is a QPI on this project, works heavily in standardizing imaging data. There is data to save omics data. But what if you have the normal, everyday wet lab data, for which you also need to have a research data management establish. We aimed, or our aims were, when we want to fulfill our goals, what we need to do is we envisioned that we wanted to have one common. Yeah, we work with, as I say, mostly wet lab scientists, no computer experts. So we wanted to have them just one tool that provides primary data and metadata. We wanted to have an automatic way of extracting metadata, so that nobody has to go back after 3 years essentially to assign it to the primary data. We want to have an automatic way of pooling it for long-term storage. And then we would like to have ideally a behind-the-scenes alignment to ontologies and perhaps to semantic knowledge bases. And particularly the first 3 steps, but then also 4 and 5, are meant so that scientists have minimal distraction from their real scientific work. Obviously, it's not needed or like carrying codes to Newcastle, but I wanted to say where this value of providing and handling data that way comes from. That is, if you are able to associate concepts or provide relationships between concepts, as for example shown here in Wikidata for a short and small part that relates around proteins, medications, active sites, binding sites, mechanisms of actions, and so on. So the goals that we have, we wanted to standardize the research data management workflow. And our INF project by the Rektorat at Heinrich-Heine University was chosen to be one of the, as they called it Leuchtturmprojekte that should have an influence on other science parts as well, including metadata and long-term storage. We wanted to adopt existing tools so that we mostly focus on what we get out at the end and do not have to start from scratch with everything. And at some point it turned up we had to develop a additional software component to support our fair research data management. I will go through these 3 parts. In the first part, eLabFTW is at the heart. For us, this seems to be the electronic lab book of choice because we address scientists in the fields of life science, less so in the actual chemistry field, but in our case, life science field. And in our case, we use it to, on the one hand, store documentation on experiments and results. And on the other hand, and this is something I need to speak about a bit more, we prepared so-called annotated protocols and materials and methods that then can be adapted or need to be adapted to become experiment documentation. This is where we want to go. So we would like to come to a point that we have data ready according to FAIR principles to be used, for example, and found again for manuscript writing, to be connected to other registries, but also to be able to either have it for publications ready or upload data as required for publications or archival. And what we needed to do primarily was we had to develop these either protocols, then they have more of the type of standard operation procedures, or what we call materials and methods parts with these so-called markups or annotations, as I will introduce to you. And we developed LISTER as a metadata extractor. And these 2 together will then allow an automated metadata extractions. And we think that this is very important because it provides less effort for users, it is less error prone. And of course, you can then get the data as required, for example, for yourself as an Excel sheet or for handling machine handling as in a JSON format. So how do we adapt then the tools? Well, as I said, we wanted to use an established technology stack, and the university had in place and uses DSpace as an open source, open access data repository software, which at the moment has been used to by hand upload data and metadata to provide it to the public. But we managed by now and are in the process to enable custom metadata search in it. We also had to do technical issues together with our research computing center, such that we all of a sudden have much larger file sizes and much larger repository file limits. We work with RDF as the standard for modeling interlinked and interoperable metadata. But what is more important from our side is that we adopted the so-called ESA model. I will mention it briefly in a minute, as a metadata framework for modeling experiments in life sciences. And as I said, we relied on eLabFTW because our university early on made the decision that this is the electronic lab book of choice. So why the ESA model? Well, it provides, in my view, a very clear how do you say, structuring of the basic concepts that are related to storing data in life sciences. There's the top level, you have this investigation level where the project context is described. An investigation can contain multiple studies. Here you contextualize information on one or more assays. And then the assay describes the actual measurements, including the description of what was done. When you then go to eLabFTW, you can see that you can map these terms very well with the eLabFTW terms and categories of system, study, stays, and experiment. We figured, however, that it would also be valuable to have yet another item, which is a project. A project could mimic, for example, third-party funding projects, grant monies, and it reflects that a system that you are working on, you can also think in terms of a system of a scientific question or hypothesis, that this often is basically finds its way into multiple projects. Then, usually to a project, a certain study is associated, one or more. Studies would then be tasks or goals. Then we have these protocols, materials and methods part I have to talk about. And we introduced such as a container, what we call publication part, which then collects which experiments at the end were actually published. And this helps us a lot to provide such data, for example, if you want to upload it to publications. So, and with this hierarchy, if you wish, we can now ask questions such as which studies, experiments belong to one system or a project or a study, as I said, for reports, for example, but also if you want to prepare publications. And on the other way around, which studies, experiments belong to a publication, such that later on, if, for example, a question comes or if you have to provide data, after it gets accepted, that you can then kind of dig out this data in a quick way. And this works in that we set up these systems with links in between. So we have the enzyme project that links to multiple studies. Studies themselves also link to systems. Experiments leads to studies. And then publications at the end are the container classes from which one can then get information on linked experiments. And from there, you can move backwards if you wish, such that if you have the information on which experiment entered into a publication or a thesis, you can always find to which system does it belong, which study, which project, and so on. And for that, we make use of the possibility to provide linked items in eLabFTW. And as I said, we make use of these annotated materials and methods parts. So how do we do the link in eLabFTW? As a example case for publications, we set up for publication, but in the same manner also for all other items or categories that you see here, such, if you wish, title pages. They need to be filled once, but then contain important, I would say, very top-level metadata information. We then defined a list of texts, and Nicolas Carpi is here, but unless I'm not mistaken, this list of text is a list as it is. There's no hierarchy in it. So we set up an initial hierarchy ourselves by using numbers, which helps us a lot of identifying links when using the search functionality within eLabFTW. And as I said, we can provide linked items with respect to publication. To make the user do all this, we provide for each of these categories essentially rules that have to be followed when you work with such an item. So this is the list of texts, just as an example from us, but you can see already that if you search up here, you can then identify easily, for example, in which part of our lab did the work was done, which systems, number 3 would be methods, and so on. In addition, as I said, we had to define this hierarchy of systems, projects, studies. And this is something that one needs to do once. But from what we learned is that this really pays off. It requires a person that has an overview of the work in the respective working group, for example, but later then it is very easy for users essentially to adapt this to their needs. I should say that these definitions should be well thought through, because they at the end determine the entire, let's say, hierarchy that we have then in eLabFTW. This is something that you have to prepare. The part on the right side is what you then have to work on as a scientist. You have to essentially describe your experiments and store your data or link your data to the respective experiments. So now comes the question, why annotated materials and methods templates? And what I thought was, if somebody writes such an experiment description, how are we going to extract later metadata in terms of formats that you wish? And we also wanted to have something that allows us to easily come to a textual description, because my view is that if experiment descriptions in eLabFTW are done very well, then later it should not be too difficult to kind of reformat them to get a textual description, for example, for a publication or thesis. And what we came up with was a markup and annotation language, which to some extent draws on what many people know from HTML type markup, in the sense that we can encode key value pairs here. We have different types of ways of how we handle these keys, either as comments that vanish or comments that can retain in the text, but we can also have formats where we have measures, units, values, and keys. And I wanted to have this because even for such a complex term here, or like terms that are in the sentence, it still allows one to read human readable the experiment description. Yeah, and that was important for us. And on the other hand, this type of markup allows us later to parse this information and extract the essential information that we require as metadata. So 2 ways. Yeah, I should say again, what one has to do as a group is one has to define, for example, these materials and methods parts. In our case, these are kind of condensed text as they could or would appear in publications or theses. But as I said before, you can make this also SOPs, standard operation procedures, which are then more detailed, have perhaps more steps. We store them in the database of eLabFTW. We tag them accordingly so that we can be more easily found. And the idea is that such annotated materials and methods will act as templates whenever you do an experiment, and the user or scientist then only has to adapt the markups. Of course, he or she can extend the description or also shorten the description. We have possibilities of having conditionals in. We have possibilities of having repetitions of steps in. And according to what actually has been done, this needs to be adapted. But from our experience, what it says is that it is much easier for a user to have such a template to start with than having to write everything from scratch every time. Yeah, and finally, we developed a software component that we called LISTER, Life Science Experiment Metadata Parser. And the idea here is, of course, that if you have such an annotated experiment description, then you would like to come to metadata in a standardized format or this textual description. And this is what we get for this example as in a JSON file or as a better human readable Excel sheet. And as you can see on both sides, left and right, we are able to keep a certain, let's say, section setting here, because we pass also the paragraph numbers that allows us to structure and organize data internally better. We have keys, we have values, sometimes measures, sometimes units. Yeah, and what is left then at that point? Work in progress, as it says here. The primary and metadata are stored in DSpace. This is what we currently are doing. And we have to customize our DSpace solution for it such that we can essentially search through LISTER metadata and catalog the data the primary data based on its metadata. Yeah, and then as a next, I think, interesting point for people that need to use such a system, this would be, for example, the clean text after parsing it through LISTER that, in my view, could be more or less directly used for a description in a thesis, if the experiment documentation is accurate and complete, of course. So which goals did we reach? We developed a workflow by adapting eLabFTW. Adapting in this case means setting up a defined hierarchy that then allows to provide links between certain items that mimic, if you want, the ESA model. And that helps us later in extracting perhaps not only metadata directly related to an experiment, but also associated with the study question, on the project information, but also on the system that is being worked on. We structured eLabFTW. As I said, this is something that requires some input of a research group, for example. And from our experience, it needs to be done at the level of a research group. I think it will be very difficult to kind of transfer materials and methods parts to other groups because there may be different ways of how to do certain experiments. You have to define annotated materials and methods parts, as I say. You have to extend, in our case, DSpace as a long-term storage. In other cases, this may be different. And we have the LISTER software that works together with all these components and provides metadata, combines it with the primary data, and then sends it to research computing for long-term storage in our case. Future work is, we still have to work better on the extraction of tables. If somebody has a question, Fatoni can answer the why here. We have to think in terms of how we handle higher level of metadata. We have to address the question. This is yet another, let's say, reorganization of science that you have to do. How do you resolve local server file paths that link primary data to your entries in eLabFTW? And I think this requires, on the long run, that data becomes much more centralized, stored here at HHU as it is at the moment. We want to identify and develop a mechanism for intermediate and long-term storage. We are at the level of intermediate storage, long-term storage is to come. And particularly that we identify how we can gather scientific data from different labs into this unified location. Yeah, and with this, as I said before, I would like to thank Fatoni Musiyafa, who has been the head behind all this, the CPC lab members in our group that provided a lot of feedback, and we had several rounds of optimization until we arrived at the system that we have. Stephanie White-Campetas as a co-PI on our project. As I said, kind of she's more looking into research data management of imaging data. Lutz Schmidt as the speaker of the SFB and SFB members for early and valuable feedback. We are happy that we have a research data management competence center now that reaches from the administration over the SIM, the library, and then to us. And Dirk Fleischer is the coordinator here. And then finally, Sebastian Mantenberg Zulauf's group. I also saw Nina Kniprath in the audience here. I just mentioned the department leaders at SIM, and we are very grateful for this collaboration as well. Yeah, and this is all I wanted to say. Thank you for your attention.